I was looking at, so the question is, so they're applying these biosolids to the fields, and the question is now, are those transporting into the plants? And so this is really a story about lignin and about plants, because in order for us to look at release these microplastics from the plants, we basically have to destroy the plant, get rid of all of this organic matter. And this is a huge, huge challenge. By the way, I'm a science teacher out in Woodward High School in Oklahoma. Also have an interest in this because I have cattle. And by the way, this wouldn't necessarily just apply to Bermuda hay. We're interested in all plants. Just acknowledgements. The previous one did acknowledgements, and same set of people to acknowledge. Well, anyway, so our question really is, does the application of biosolids in hay fields result in the uptake of microplastics into hay plant tissues that would be consumed by grass foragers? There's a small set of studies, and it's growing pretty fast, that have found evidence for plants to uptake microplastics into the soil, into the roots, and then into the shoots, so that above the ground. And what's interesting is most of these studies are really in the lab, hydroponics, feeding the microplastics in water. So I mean, this is kind of an artificial setup, and what's cool is Dr. Nanny will be looking at actual grass in a field. And that's pretty exciting, but it's a hard thing to do. Application of the biosolids to hay fields may increase microplastics, and I think we just saw that, maybe I shouldn't have said may, I said it does increase microplastics out in the fields, because certainly there's what was a 9-fold difference between the control and the hay fields.
For the light fraction.
Yeah, and so this slide is kind of showing the broad range of issues that we're going to have with microplastics. So first of all, soils, we've got soil composites, agricultural equipment, artificial turf, polymer-based agrochemicals, wastewater treatment, so the biosolids is here, irrigation water. Not so much here in Oklahoma, but other places where they do more of the garden type plants, they use a lot of agricultural films, so plastics to cover surface of soil. And we have a lot of interaction with co-contaminants. So one thing about the microplastics is they can serve as a substrate that carries other things. Particularly interested, so the literature is particularly interested in some of the heavy metals. Cadmium in particular seems to transport with microplastics. But it's copper, arsenic, or other issues that people are worried about. The plastic additives, so the phthalates and the bisphenol, things like this, also might be a concern. And over here on the other side, physical effects affecting this whole soil chemistry, just the organic matter, just the whole dynamic of the soil. I mean, I think one thing that's interesting about your columns is why is the particles evenly distributed down through it? I mean, what's happening to the dynamics of the particles as they move through the soil? Just changing the porosity and the density and so forth of soil. So I mean, we're adding these things. What does it do to water movement through the soils? So there's a wide range of issues. In terms of the plant tissue, we would expect to find most of the microplastics down in the root system. Transport up through the plant seems particularly dependent on the transpiration pole. So plants that are growing really fast in sunlight, putting on a lot of growth, we would expect to see those microplastics moving through them fairly rapidly. More mature plants, maybe not so much, because we may not have that transpiration pool that's quite as strong coming up through the plant. So moving through the xylem, pulling those microplastics up into the plant tissue. We might also be getting some microplastics moving into the plant above the ground. Just airborne, so the atmospheric deposition may be also directly on the plant, absorbing into the plant, going through the stomata of the plant, things like that. But anyway, this gives you an idea of some of the issues. Well, anyway, the problem is nature finds it hard to digest cellulose and lignin. It's not easy. Ruminants use, like cattle and so forth, these multi-chambered stomachs that they use. They have this whole digestion system that's designed to do it. But by the way, this is on my farm, and these are some cow manure on the field. I mean, if you actually pull up this cow manure, by the way, I was an agriculture major. I've actually fingered around in cow manure in the lab at different times. It's still filled with intact plant in there. And so even with what they do, and by the way, the cow here was, so you know, it's a cow and the calf here, they lay in the fields a lot just chewing the ruminant. I mean, they actually reprocess what they eat, trying to break this all down to get energy. So the dung is still rich in complex carbohydrates, and it's a food source for lots of things. I love the dung beetles. I've got dung beetles everywhere. They're rolling balls to and fro, dung, and it's a lot of fun just watching that. Of course, the insects, the flies, and so forth. Amazingly enough, a lot of animals, including my dog. My dog likes little plops from the calves. I think it's still a little milky, and so loves to eat that to my chagrin. Oops, wrong direction. So anyway, traditionally, some of the ways in which we try to break down lignin: oxidation with acids, alkaline digestion. I had a student that wanted to make transparent wood, so to remove all the lignin from the wood so you could see through it. So he hit it with acids, he hit it with sodium hydroxide for hours on end. Finally got some transparent wood, but anyway, it takes a long time through these oxidation processes to really break down this lignin. The cows are doing a lot of enzymatic digestion with the help of all their microbial friends. So we looked at enzymatic digestion, we did cellulase digestion, but we really weren't happy with that result. We didn't get a lot of breakdown from it. Now there's a lot of tricks to it, and we didn't have time to play through all of that, but it wasn't, at least for us in our initial attempt, wasn't very effective. But Dr. Nanny and Joe have been working on these ionic fluids. This worked really, really well. And so this seems to be a very promising place to start, because the samples that we processed with the ionic fluids were broken down. There was very little identifiable plant material after doing that. And so that's very, very promising. I think at this point with that, we can start to look at doing that. So in terms of the process, you take plant materials, you get them dry. We have to have them dry because the ionic fluids and water are going to be a problem together. They're both really polar, right? And we're just going to have too many interactions. We still do the oxidation, in this case with the H2O2 and then with some iron chloride and nitric acid. Dried and weighed, trying to get the densities. And Dr. Nanny said we didn't actually do the sodium chloride density extraction, but this we can do density extractions with this, dry in the oven and weigh. And then we just put it in our ionic fluid. I'm not going to say the name of it because it's a big, long name, but the abbreviation here is EMIM-OAC. So it's an acetate type stuff. And anyway, we put it in for, in our case, we did 48 hours at 95 degrees Celsius in an hot oil bath, stirring the thing, and just keep going. And by the way, just a note, polyethylene melting temperatures around 105. So keep it just below that. This is some pictures of just some of the work that we did. So here we have a plant from the sample fields that was talked about earlier. Oxidation. This is in the hot oil bath. Got 4 sample bottles here. You'll notice really dark black. By the end of it, this will turn kind of a more reddish color. This was a dark brown. These were the roots. We had much less root material. So Bermuda grass doesn't really put on an extensive root system. So we didn't have a whole lot of roots. Trying to see. And this is an example of, we got a lot of breakdown. When we first did it with just the cellulase, our filter looked like a haystack out in the field. We still had a whole bunch of organic matter just piled on top of the filter. But this got a really good result. And I think if we go longer, we could probably get that down. Here we were doing the hydrolysis in an incubator on a rocking table. And so this is kind of the process we're looking at. So sample preparation, just getting it cut up into small little pieces. The oxidation, do that 2 times. Do the ionic fluid to help decompose that lignin. And we may look at doing a hydrolysis with the cellulase to kind of polish everything off, get it to hopefully break down what remains into it. And then the non-red staining, and then the fluorescence with the digital microscopy. And now one of the issues we're going to have is with the plant microplastics is we're probably looking at 5 microns and smaller. We just simply have a physical barrier. Humans, we get a lot of microplastics because we like to consume everything, right? We drink from all different sources. We seem fairly indiscriminate. Plants kind of sit in one place, same environment, same water source, and they're producing, they're producing their own food, and so that epidermis on the plants is an effective barrier where we're just, putting things down our gullet that have plastics in it. And so that's one kind of difference. And so the sizing is going to be an issue with plants. So we should be able to see things, again, down to that 1 micron range, but we do expect that we're going to be looking at those nano-sized microplastics also in the plant tissue, particularly those that go up into the shoots. Again, it's a lift to move a 5 micron, but those, get down to the billionths of a meter, they may move readily up through the plant. This is just showing some of the shoots cut up. I don't know why I said shoots twice, but and some of the roots. But we process them down. When Dr. Nanny told me I wanted a hammer mill because I was an ag major, and so we would grind this into dust to do nutritional analysis and stuff. But we didn't want to necessarily damage the microplastics themselves in the process. But we get it down to a nice small size to help with that breakdown of the microplastics. And then this is just showing, again, and I didn't put in a haystack picture, I should have. But here we're seeing the shoots after staining. Just, we have a little bit of the grass here. There's like just a few little pieces left. Likewise with the roots, we have a little bit of organic here. But we've really broke down all of that organic matter, and so
you might want to explain what all those white fragments are.
Oh, the unfortunate thing maybe with the process is we're doing a number of filtrations, and so this is the broken up filter. So we move the filter each time because it's going to have microplastics attached, and we just simply break it up, move it to the next filtration. And so ideas for maybe streamlining to not have quite so many filter fragments may be good as well, because they do get in the way. Oops. So anyway, these are Dr. Nanny's next steps. He's wanting to get this again to that standardized, high throughput method so that we can begin to analyze these microplastics in the plant material itself. And that's a big challenge. And again, down on this microscopy end, we're definitely going to miss some because we just aren't going to be able to see the smaller pieces. But we do expect to see, again, 5 microns down to that 1 micron range. We do expect to see something there as well. So it depends a little bit on the type of plant and so forth. Bigger plants can move bigger things. And I just have a reference slide. And that closes it.
Any questions from the audience? Yes, Ting.
For your sample, okay, did you choosing different type of, like different age of the plants? And will you also choose, because you mentioned that the age limits mature plants and the immature like growing plants, rapid growing plants may have a different consumption of their microplastics, as well as the different latitude of their plants, like leaf on the bottom versus leaf on the top. Have you ever choosing different samples?
We didn't, so we just simply separated roots from shoots. And then we mixed to kind of have an even sample. Because the shoots, of course, we got stem and blades, the flowering parts and stuff for panicles, stuff like that.
So these samples were collected from the fields we took core soils, soil cores from, and the soil and the plants were collected right before harvesting the Bermuda grass for hay. The idea is wanting to see, that hay is then used to feed cattle. So the whole focus of this overall big study with the geological survey is, applying biosolids, microplastics, PFASs, other things. How do they end up into the plant, and do they get into the beef cattle? So, that what you proposed was not a research question, but I mean it is an important question. But we're just taking an opportunity here to look at these grass samples.
And again, it's not just the cow, because we're going to have a lot of the grass still pass through the cow and be processed by bacteria, fungi, all the insects, and my dog, unfortunately.